Hello, my name is Mike Ward. I'm the Global Director of Content at Scrip, The Pink Sheet and In Vivo. We're here at the Biotech Showcase 2018 meeting, which runs parallel with JP Morgan Healthcare Conference, which is the traditional kickoff for the pharmaceutical industry, where pharmaceutical companies, investors, biotech companies all get together to sort of discuss some of the issues that they face, to also share progress on what they're doing, and also look for sort of potential deals, whether that's investors or companies getting together. There are lots of different areas that people are looking at. One of the interesting spaces is in autoimmune. There's not a lot being done on it because a lot of autoimmune disease is about actually suppressing the sort of immunity. But I'm joined by Jesús Martin-Garcia, who's the co-founder and CEO of GeNeuro, which is a publicly traded Swiss biotech company in the autoimmune space.
Absolutely.
So thanks for joining us.
Pleasure, Mike.
So I mentioned that the approach normally about autoimmune is to try and actually sort of suppress the immunity.
Absolutely.
But you guys have got a different approach.
Absolutely. Today, against autoimmune diseases, what you try to do is to diminish the immune reaction to a problem you don't actually know.
Yeah.
And we believe we have an insight into what this problem could be, based on the work that was done in INSERM and In situ milieu already 25 years ago. And what we're trying is to go with drugs that could block causal factors that are driving these autoimmune diseases. So what are these, in fact? We believe that a lot of these autoimmune diseases are driven by proteins that we are making from viral genes which are in the human DNA. In fact,
these are genes that are embedded there.
Absolutely. It's actually 8% of the human genome is made of genes which are left there by infections that happened to our ancestors. And this is an ongoing process today. There's kids that are born with some genes with AIDS. And this is very well suppressed in our genome, otherwise we wouldn't be walking. Yeah. But in some circumstances, most importantly, where an external virus comes replicating into a cell, it allows its cousins within our DNA to express proteins. And some of these proteins may be toxic. And those are the proteins we're looking at. We're in particular looking at one of the 24 families, which is the human endogenous retrovirus W family, HERV-W. And the envelope protein of this family is found in very high quantities in the plaques of MS patients, as well in the pancreas of type 1D patients. And what we've developed over the years is an understanding what this protein is doing in the brain of patients. What it's actually doing is twofold, which are very relevant for multiple sclerosis. On the one hand, they are activating innate immunity via the TLR4 receptor. They're activating microglia and provoking the release of pro-inflammatory cytokines. So those viral proteins in the brain, which are made by our own DNA, are contributing to inflammation. But as importantly, they may also be part of the mystery of why is the repair system of the brain blocked in MS patients. You know that whenever we have an insult to myelin, we have the cells called oligodendrocyte precursor cells, OPCs, that come to the site of the lesion and remyelinate to protect the axons. In MS patients, those cells migrate but don't remyelinate. And what several academic groups have found is that in fact, in the presence of this protein, they stop remyelinating. So we hope we have an antibody that neutralizes this protein, and we hope to see two effects. First effect is to lower the inflammation. And the second effect, I would say, almost as or even more importantly, is to restart the remyelination process and therefore contributing to slowing down or stopping neurodegeneration in patients. So that's our goal.
So what proof of principle have you already achieved with this approach?
We had a phase 2A on progressive patients, which was just on 10 patients, was very successful. That led to the partnership with Servier. And today we're finishing a 270 patient phase 2B in MS. We had preliminary results after 6 months already in October. We presented at MS Paris, where we showed that there was a benefit in inflammation more towards the end of the period, not on the whole period. But more importantly, we show first signs of remyelination in patients at already 24 weeks.
So those were the endpoints then?
Absolutely. The primary was to get a limit enhanced. We didn't have that one. We're doing clinical research in the most noble sense of the term. In the sense we are opening a new area, and in the inflammation area we had an effect, but it looked like a delayed onset of action because the benefit we were bringing was concentrated at the end of the 6-month period. So we have to see now at 48 weeks at the end of the year whether that effect was maintained, sustained. But almost as importantly, we started seeing signs of remyelination in patients as we had an increase in the empty signal, which is the best surrogate we have today for remyelination. Obviously, we're very much looking forward to seeing that at the end of the year.
Yes, yeah, sure. So you sort of say that you're partnered with
Servier
on that. So could you just sort of outline the details of that deal, because you still retain some of the rights?
Absolutely. What we did with Servier is is a fantastic partner because it's a private, very science-focused, long-term focused company that's been a fantastic partner for us. And what we did is we have a deal in which Servier has an option to license the rights of our compound at the end of phase 2B, worldwide rights ex-US Japan.
Okay.
So we've kept the US and the Japan rights, and for that they are funding the development in the MS, both in the US and Europe.
So at the end of phase 2B, if it all works, they will then take the program on themselves? Is that how it works?
That they would take the license on the compound, if obviously they wish, it's their choice. And if they do that, they will fund the continuation of the development in MS, including the US, in which we have retained the rights.
Okay, okay. And your plan for that would be to what? Find a US partner or develop it yourself?
Ultimately, we're very open to that. We don't have a real pressure, since Servier would fund, if they take the license, would fund the development, including the United States. So we don't have a real pressure, but obviously, for many practical reasons that we all know, eventually these are things that we're discussing, that we will be discussing with potential partners in the field.
So these ancestral viral proteins, are they involved in any other diseases that you're targeting?
Absolutely. There are 26 families of these retroviral genes into our genome. We have an ongoing study in type 1 diabetes right now, which just completed recruitment this week, and we expect results from that in September of this year. This is really very exciting and interesting trial.
And what is that target?
What we're looking at is whether we can preserve the capacity of those patients to continue to produce insulin when it's not yet down to zero. Is to preserve it, because we believe that the agent, this pathogenic protein from our genes, is actually driving the destruction of the beta cells in the pancreas. So by neutralizing it, we hope to stabilize the condition and the ability to produce insulin. But we're also working with the NIH in ALS. And this was work done by the ALS with another family, the human endogenous retrovirus K family. They had a fantastic publication in Science 2 years ago. They were working together to establish a therapeutic clinical strategy to go against that disease. And that is also extremely exciting.
And where's that in?
It's in preclinical today.
Right, okay. So when would you hope to get that into humans?
At best, I'd say sometime next year. Sometime next year in 2019. But this is very exciting. What we're basically looking at is a new way of looking at this part of our genome, which is 8% of our DNA, and which is an extremely interesting part because it's mobile genetic elements, and they are extremely diverse across ethnicities, across individuals, and we believe that they may give us part of the mystery of why does autoimmunity triggered? How is autoimmunity triggered?
So the Servier deal, presumably there was some money associated with that. So
Yes.
Do you have enough sort of finance to be able to sort of progress the programs that you've got on board?
Absolutely. Between the IPO we did in 2016 and the milestones we received with Servier, we're funded till mid-2019, which is more than enough to read all the MS trials, to read the type 1D trial, to get the ALS plan to go into the clinic, so we're really well funded. Plus we have a very solid group of shareholders with the Mérieux family, the Institut Mérieux that has been with us since inception. The co-founder of the company was late Christophe Mérieux, and with a strong Swiss investor group called Eclosion too. And of course Servier, which is also part of our equity. So we have a very, very strong shareholder base.
Right, okay. And I mean, I guess sort of you know, going forward at this meeting, you know, people are here to talk to investors or to talk to potential partners. You know, whose ear are you trying to bend?
Well, it's important for us. We're a European company based in Geneva that actually has retained US rights for our compound. So it's key for us to come here to the US and to talk to potential partners, both in pharma, both financial, and I said, to let the community know what we're doing. Because, you know, as a small European company, you tend to be very focused on what you're doing. So we're really blooming out into the United States and preparing for future steps.
Okay, great. Well, Jesús, thanks.
Real pleasure
to
meet you.
great.
Thank you
Cheers.
very much.
Bye-bye.